all. I welcome Dr. Madhavi Julakanti, and she's a professor of the upgraded Department of Anesthesiology and Critical Care, Osmania Medical College and Osmania General Hospital, Hyderabad, Telangana. She's faculty for the state and zonal conferences and has got a lot of publications in the national and international journals. And she's a team member in organ procurement of brain dead donors and also team member of liver renal transplant surgeries in Osmania General Hospital. Faculty for GRACE, that is a Global Regional Anesthesia Curricular Engagement under WFSA. Welcome, ma'am, and over to you.
Thank you, ma'am, for kind introduction. Good morning, one and all. Thank you very much to give me this opportunity to speak in South Zone regarding immunology basics. I kept this photograph because it witnessed the first kidney transplant and the first liver transplant in Osmania General Hospital. We have not witnessed the first kidney transplant which occurred in 1994. Still the brothers, siblings who were the donor and recipient, still they are surviving because of that immunology. That's what I feel. The history. Lord Ganesha. The research started, I think, through the Indian mythology, looking at Lord Ganesha's. So it started, that's what I feel. We all must have come through the library complex of Mysore Medical College, where we face the legendary surgeon Sushruta, who is in the 5th century of 5th century, he did the first nose reconstruction with autograft, but there is no evidence, there is no documentation. That's why we could not document it. We all know that in 1908, Alexis Carrel, who got the Nobel Prize, started kidney transplantation in cats, where he could see that the kidney function, the cats urinated for more than 25 days. It started like this, and when Joseph Murray, first he did the first transplant among identical twins, followed by Samuel Cohn's among the non-twin. In 68, the first heart transplant which occurred in Bombay, that was first heart transplant by Prafulla Kumar Sen. 1971, the Dajokshi and Mohan Rao, we all know that the first kidneys transplant occurred at CMC Vellore. From then onwards, we are continuing in India to do the transplantation programs. Why this immunology base and advancements were given to me by this South Zone Scientific Committee, I don't know, but they made me to read about immunology basics. It is the way related among anesthesiologists. Till now, all the sessions we were talking about anesthesia, but the immunology among us who has read about immunology basics, I don't know, but they made me read this topic, and it is so debatable among nephrologists, hepatologists who are doing continually handling the immune response which occurs among the patients after transplant and pre-transplant. Nothing but from the mammalian immune system is an extraordinary complex system. We all know that it is with when life started along with the microorganisms, so much of stressor response, this immune response is evolved. It has divided into natural immunity, which is a non-specific response. Whenever there is an injury among us or anywhere or infective process, this natural immunity, non-specific response would occur in our bodies. That adaptive immunity, which I'll be talking about, a response to a specific antigen. I'll be talking about only this adaptive because it is there with the patients who are taking who are having the transplantations from the donor. In organ transplant, the principal target of the immune response is the graft or the MHC, major histocompatibility molecules. The natural immunity is developed there with the macrophages, neutrophils, natural killer cells, cytokines, cellular receptors, and complement components. Where the adaptive is a specific antigen and confers both specificity and memory effect by T and B lymphocytes. T cells recognize the antigen in the form of peptides bound to the MHC proteins, B cells immunoglobulin receptors that can recognize the antigenic portions of the intact molecule. If we take the B cells, for example, whenever there is a change or there is involvement of B cells, which is humoral immunity, before what they used to do, we should have compulsorily ABO incompatibility test before going for transplantation. So most of the people, if they don't have, it was compulsory, they used to go for desensitization programs in the earlier, which was plasmapheresis or splenectomy or immunosuppression. Because immunosuppressants are developed, that's why we are going for this is era has become the transplantation era. We have lot of pressure in the government sector and private sector to go for more of transplants. So natural and adaptive immunity system, which are they are closely interrelated. Antigen specific T cell activation leads to the production of cytokines and chemokines, recruit components of the natural immunity, allo antibody production and CD8 positive cell mediated cytotoxicity, local tissue production. For that, local tissue production of complement appears essential for T cell activation. Whenever there is a antigen specific recognition, there is always T cell activation. I'll be dealing more of T cell activation, how it is going and having developing the cell mediated cytotoxicity. Whenever there is not our body, it will try to reject it from the body of the recipient. So how T cell activation is taking place? T cell recognition of antigen is the primary event. So the key steps to discrete the signals by interaction with T cell receptor with antigen presented as peptide by the antigen presenting cell, there should be a co-stimulatory receptors, whether ligand interaction on the T cell or APC cell surface. T cells undergo clonal expansion under the influence of mitogenic growth and differentiation factors. There are so many differentiation factors whenever the T cell will go under clonal expansion. Among that, interleukin 2. Recently also we all know that we used to, during COVID pandemic, we would go and get the interleukin 6, how much is there, based on how much response is there or how much infection is there. So like that in the transplants, interleukin 2 is more important. To act, whenever the activated T cell, they perform to induce CD8 positive cell mediated cytotoxicity, provide help for B cell antibody production. If they provide help of macrophages, so they'll have delayed tissue hypersensitivity response. So I'll be talking about in detail about MSC structure and that its function, and followed by the T cell activations and T helper cells and how the cytokines will respond. Region of highly polymorphic genes located on the short form of chromosome 6. Because of this polymorphism only, we have a lot of changes in the recipient body. The protein product of MHC are expressed on the surface of variety of cells. That means most of the cells except red blood cells. The human system is called HLA, as we all know. We all get the HLA from our parents, the pair of genes from one parent, and second is the mother or father, and the sibling will have 25% of 25% chances if we get any time the organs from our siblings. So the cell surface proteins which are there, they are the principal antigenic determinants of graft rejection. I will be speaking about only rejection and how to come out of battle the graft rejection with the immunosuppression. Organ transplanted always between MHC in identical individuals will be accepted more and more. Mismatched individual will be rejected. So MHC class 1 all nucleated cells with CD8 positive cells. MHC class 2 are macrophages and dendritic cells and CD4 T cells. So this is the two classes of MHC which are structurally little different, and their antigenic binding groove is one is closed and one is open. Peptidic length will be, amino acids are different. One is the source of antigen is intracellular and the class 2 is extracellular. Expression will be all nucleated cells of class 1 MHC, and class 2 MHC will be the B cells and macrophages and dendritic cells, where antigen presented to CD8 T cells of class 1 MHC. Class 2 MHC is CD4 T cells. These all are causing the cell lysis and cell death followed by rejection. So minor, other than major histocompatibility, there are minor transplant antigens. These also, they'll have a lot of, especially they'll have a role in bone marrow transplantation and will have graft versus host disease. Now, the mechanism of allorecognition. There is always, they try to reject, whether it is through the direct allorecognition or indirect allorecognition. Whenever the T cell, which will recognize the donor antigen presenting cell, then the leukocytes will go and they'll catch up and they'll try to reject. Normally, the indirect allorecognition is the common pathway, but the direct donor APCs or recipient APCs will be having the major role with direct allorecognition and rejection and indirect allorecognition. This, how to enter, T cell to enter to the cycle, cell cycle, there should be a T cell co-stimulation. You please see that CD8 which is adding to the MHC class 1, and there are so many co-simulators, T cell factors and inhibition of T cell, and there will be so much of secondary messengers will have to have this immunological synapse. There is a adhesion among all the factors. So if we see the CTLM, this is cytotoxic lymphocyte mediated one. Nowadays there is a lot of studies among them. So these have a immunoglobulin which is recently developed, which will have intraoperatively or preoperatively they'll be giving the immunoglobulin so that means there won't be immediate allo rejection. So in our hospital, you must be having most of the people who are giving anesthesia to transplantation perioperatively before reperfusion injury, we are all using ATGs, anti-thymocyte globulin. So before releasing the clamp, by the time it will get reperfused, if we give, the rejection will be very less. Still, there are so many investigations going on, so many investigations we do before entering into, before taking the donor and recipient. Whichever advanced, still the rejection rate is high among donors, among recipients. So T cell homing, why it is useful? Whenever there is the T cell homing in the hematopoietic system, the cell to come out of and to go, there is so much of molecular mechanisms take place. The leukocyte migration will take place with the rolling, adherence and diapeptisis. So to enter into the endothelial cell. So recently developed that if we take the investigation of endothelial cell antibody, so there are so much of research is taking place that after this endothelial cell antibody recognition, maybe the rejection rate will be lessened. Cytokine and T helper cells, how? We all know that the naive T cells, they mature and develop in the thymus. So there are Th1 and Th2. Th1, which is interleukin 2 and interferon gamma and beta, they'll all cause cell-mediated cytotoxicity, macrophage activation, they'll take the B cell help. This is the response, immune response, which is the cause for immediate rejection in the recipient by the organ tissue. So this one, the 2-1 is eosinophil mast cells and immunoglobulin E and G, they'll take, but these have transplantation tolerance. Followed by molecular mechanisms are T cell activators. Whenever there is antigen presenting cell, they are on the surface of the cell. They'll try to enter into the nucleus and call lot of biochemical molecular mechanisms to cause there is increase in the calcium, where the calcium, there the calcineurin inhibitor will have a immunosuppression at this level. Those are cyclosporins, and the 2 is the tacrolimus, which is a good immunosuppressant which is in the liver and kidney transplantations, and the rapamycin and the steroid. These all are the immunosuppressants which are which are very useful so that it won't go to allo rejection. I'll finish it soon. So how it will take, I already told that this mechanism, interleukin is the main culprit, and after T cell activation, there is clonal expansion of alloreactive cells, effector mechanisms, followed by alloantibody production, which will cause the death and delayed tissue hypersensitivity, followed by apoptosis and induction of cell lysis. So, in the end, my summary is organ transplantation is a form of adoptive immunity. As anesthesiologists, not only nephrologists or hepatologists who are dealing with this immunology basis, we also a part, because so many cases after rejection or chronic rejection, they'll be coming and they'll coming to us for anesthesia. Lastly, you all know that the great leader who is a motivator, and she is the dynamic leader whom we lost after renal transplantation. Thank you so much for giving me the opportunity. Thank you.